Hello, check. Hello.
Good evening, everybody. It's a pleasure to welcome you all for the 14th FM Oration, the Fieldhouse-Myers Oration. We'll begin with the program straight away. I request Dr. Priya Abraham to give the welcome address. Dr. Priya.
It's my privilege to welcome our Vice Principal, Dr. Jacob John, our speaker for today, Professor Justin Bahl, my colleagues from the Department of Virology. I'm not seeing anybody else outside our department. I hope they will trickle in. And this is a very important annual event, the Jeanette Myers Oration that we have been holding almost every year. Of course, during the pandemic, it took a hit. And we have always maintained the tradition of inviting a virologist of eminence, a global expert. And today we have a person who truly befits that expectation, Professor Justin Bahl. He is a global expert in infectious disease ecology, evolution, and epidemiology based in the University of Georgia, in the state of Georgia in the USA. I will not steal the thunder from our Vice Principal, who will be giving us a more detailed account of our orator for today, and I would request our Vice Principal to kindly say a few words.
Stage, so it's easier to see. Dear Dr. Justin, thank you for coming to be here with us at CNC. It's truly a pleasure to be here today. Actually, the principal would have been here, but for the fact that he had to leave for another meeting and he's not here in town today. So, welcome everybody. I hope more people are going to be able to join in. I've had the privilege of listening in to a few of the orations before, and it's really been something that's really enriching. And today, we're really privileged to have you here with us, Dr. Justin. And Dr. Justin, as you all know, is a very accomplished scientist. He's also a person who has fostered many researchers during his career and his work in Georgia. In today's world where we are working, constantly being challenged with so many organisms, so many problems that actually are so rampant. They come out so suddenly and we need to find solutions for them. No longer is it sufficient to know about the pulse. We do need to know about what's happening in the environment. We need to know about the host mechanisms, and we need to be able to really agile about how we deal with it. So the virologists working along with the veterinarians and the epidemiologists, that's really what you've been doing and fostering. So we're really grateful to you for coming here and being with us, and we hope that you will be, the talk today will be really useful and encourage all of us to be able to look at a similar multidisciplinary way of functioning. Thank you for coming here, and I hope you enjoy your stay here. But again, I will come back to talking a little bit later. Over to you, sir, for your talk. This is the next. We will not take away your time. Thank you for coming.
So I've been given the privilege of introducing Professor Justin Bahl. Justin's an old friend. I knew him from many years ago, and I'm really glad that he found the time to come here. So he's a tenured professor at the University of Georgia and serves in multiple departments, the departments of infectious disease, epidemiology, biostatistics, and importantly, the scientific director of the Center for Applied Pathogen Epidemiology. He has completed his BSc with honors at the University of Toronto in 2001 and earned his PhD in molecular systematics and evolution from the University of Hong Kong in 2007. He has worked with numerous reputed groups and he has over 100 peer-reviewed publications. His research has focused on genomic epidemiology, viral evolution, and outbreak response, and shaping global surveillance systems and vaccine strategies. He has served as on numerous scientific committees, the WHO, OIE, FAO Working Group on Influenza Nomenclature, and regularly reviews grants for leading international granting agencies. It's a real privilege to have Justin here, and good to see you after all these years. A warm welcome to Justin. Over to you.
Great. Well, thank you so much. It's always interesting to hear somebody read my history. Oh, okay. Read my history when I'm actually way more humbled being here. I feel like every U.S. official that has supported the work that's happened here needs to actually see what you guys are doing. It really is quite amazing. And the other thing I want to emphasize is that I think a lot of grants that have gone into public health have actually damaged public health because they've moved away from the public. And your engagement with the community allows you to affect real healthy change, which is what the entire center that I've developed is about. It's actually taking epidemiology and applying it to make people healthier. So it's really wonderful being here, and I'm learning so much in the very short time that I've been here so far, and I look forward to speaking to everybody throughout my stay. So today I'm going to talk about cross-scale phylodynamics. I'm going to focus on respiratory viruses. I am going to jump around a little bit between different viruses, but I hope you're going to recognize a theme. I'm going to start slowly, and then we're going to go really fast. Okay, so hopefully the early part will give us the foundation to understand everything that I'm going to talk about. So first, let's define the word epidemiology. I like looking at Latin roots of words because it forces you to really understand what that word means. So epidemiology comes from the Latin of epi, demos, and logos, okay? And that is the epi meaning upon, demos people, and logos study. So it's the study of what is upon people. I specialize in molecular epidemiology, and this is where we are using things like genomic data and other molecular information to learn about what is upon people, okay? This is a fairly new branch of epidemiology and emerges this, the field of epidemiology with molecular biology. Specifically, I'm working on infectious disease molecular epidemiology. So now we're using genetic data to identify and unravel the source, find out where reservoirs of disease might be, and trace its circulation pattern and transmission patterns. Through the last few years that we've seen emerging diseases, spillover events, genomic sequencing and the analysis of genomic sequence data has become central to understanding the disease dynamics of especially RNA viruses. SARS-CoV-2 this is the most well-studied infectious disease that we've ever encountered. We've generated millions and millions of genomic sequences, and now we've got the challenge of coming up with the methods and the interpretations of analyses for a scale of data that we've never even, it wasn't even a reasonable prospect before SARS-CoV-2 happened. And I am going to talk a little bit about the Delta epidemic, which was first identified through sequencing here in India. But before I get to that, let's start, like I said, we're going to start slowly. So what is a virus? My definition may be different than other people's definition here. Specifically, my definition focuses on its life cycle. So it is an infectious, obligate intracellular parasite that comprised of genetic material surrounded by a protein coat or membrane. It is an obligate intracellular parasite. Many people will consider the virus to just be the particle. And let me see if I get the... Okay, many people would just consider this particle to be the virus. But I actually consider the virus to be a whole organism that includes the infected cell and the virion. So the virion is just that particle. And the genetics within this virion are restricted, that it has to be able to replicate within a specific host cell. If you had, say, some lentils for lunch or onions, you've been exposed to onion viruses. They don't replicate in you, they don't cause any disease in you, they are restricted to being able to replicate in onion cells. So the genetics restrict what we see in that, or where it can replicate and where it can be successful. So if we're considering a influenza virus that replicates in humans, when I study the genetics within this virus, or within this virion, I am learning about the infected host, because this is an entire organism, okay? So we have an infected cell and that actual virion, and that infected cell really does restrict what we see in the genetics. I think we're also in this age of pandemics. I've heard it called the pandemic age, but we're certainly in an age where we have had such a huge impact on our environment that we see a number of new epidemics emerging. We can go to 2009 when we had the H1N1 pandemic, and since then we've had measles reemerging, we've had MERS come up, we've had Zika virus, and of course leading up to SARS-CoV-2. And that's just a small sampling. So we're seeing a lot more new emerging diseases come out. And we really do lean on genomic sequencing to try and understand these different viruses. The other element of this modern era is I think we're moving beyond the individual and really trying to view things as a system. So even in from the scale of a single individual, maybe that biomedical scale, the doctors here, you may be working with a single patient. If you study the microbiome and the microvirome of that person, you can learn about where they've been, what they've been exposed to, what sort of environment do they live in, right? Do they live close to industry? Have they traveled? You can learn a lot about that individual and understanding the system that they exist in that may be posing a risk to them. And then scaling up, when we look at emerging diseases, we really need to start understanding things in terms of an ecological framework. My goal as a scientist is try and bridge these different scales, going from that individual to that ecosystem. And I do that using genomic data. And this is a typical example or a typical model for the emergence of pandemic influenza, but I think it could be applied to other diseases. So we have viruses that circulate in the natural environment. These animals move, they migrate, and they somehow interact with domestic animals. When that spillover to domestic animals happens, that's when we have an increased risk at that individual of acquiring this new disease. And that might interact with other animals and then cause pandemics. So I think understanding what this system looks like and all the different pathogens that might actually cross that domain provides us with the key understanding that we need to stop future pandemics. And if we look at how H1N1 emerged in 2009, this is the same for the SARS-CoV-2 pandemic, 2019, that we had these viruses that were moving between, wild and domestic animals. The H1 for that influenza pandemic goes all the way back to the 1918 virus. It shares a common ancestor with that and was circulating in swine since then. We had spillover from human influenza getting into the swine population. We had avian viruses coming from the wild animal population moving into Eurasian swine and circulating for a long time. And then they all jumped over at some point. These Eurasian genes managed to get into the North American swine population, likely in Mexico. That mixed with these viruses that had human origin genes and other avian origin genes to start a new pandemic. That period was about I guess if we go all the way back to the late 70s for that first introduction, is how long it took for that virus to actually emerge. If we look, some of the more recent transmission events between these populations, it really was about a 20-year period before that virus actually emerged. So it was circulating in swine and able to mix for a long time. Okay, so that's just a bit of my soapbox as well, that understanding the ecological system is going to allow us to better respond to pandemics. And where I said that this is my primary research area, many people, just to redefine or reaffirm what that scale might be, many people, medical doctors here, will be looking at that biomedical scale. So you're interested in that individual that shows up sick. As an epidemiologist, we would be interested in the population scale. So here, the unit that we're interested in is not an individual and the pathogen, it's transmission between individuals, okay? And then an ecological scale is where we're looking at transmission between populations. This could be multiple human populations that are geographically spread. This could be spillover from wild animal populations or domestic animals. And as this virus moves between them, this is the ecological scale. An ecological study is an epidemiological study where the unit of interest is a population rather than individuals within a population. Okay, so there are parallels between studying what happens within a population and what happens between them. Okay, and because I'm studying genetic data, I focus on evolution. All right, genetic data is the material that evolution acts upon. We need genetic diversity for evolution to select what might be successful or not. So understanding those processes that generate genetic diversity is important. And that will help me understand why I'm seeing the different viruses that I do see. So there's 4 basic mechanisms: mutation, migration, genetic drift, and natural selection. Most people will just think about natural selection in terms of evolution, but there are these other mechanisms as well. And then when we sequence viruses and we build a phylogenetic tree, and I will talk a little bit more about phylogenetic trees, but as we build the phylogenetic tree, we're creating a hypothesis of its evolutionary history. And we're observing genetic diversity at a single point in time, and we want to understand what was the history that generated that genetic diversity. So we want to understand how all these different processes might have mixed together. If I take just H3 influenza viruses that are collected from humans, from wild birds, and from domestic animals, and build a phylogenetic tree, they look drastically different. This is a typical influenza phylogenetic tree. It has this ladder-like phylogeny, as we describe it. It's very spindly. If you imagine a very, very thin tree with few branches. That's what we have with human influenza. We have a very strong immune selection. We have vaccines. We don't generate much diversity of influenza viruses. Most of it is eliminated during an epidemic. So we end up with this tree that's very long and spindly. With wild birds, the time scale goes back hundreds of years, and we have this big bushy tree, right, with these big branches that come back hundreds of years, and the tree just sort of grows at a very regular pace. When we get to poultry, we have sort of a mix between these two. We have very short branches that are connected by long branches, almost like a bunch of palm trees that come together at a single point. Okay? So you have this long branch and then an explosion of leaves and tips. That's an epidemic outbreak that we can see. So it might be getting into a chicken farm and spreading through all these chickens. We can see these different processes at play just by building this phylogenetic tree and looking at how it might look different as a result of those different processes. This brings us to a field called phylodynamics. This is a field that was defined in 2004. And this is where we could take rapid evolution of pathogens, and I'm emphasizing RNA viruses, although it has been shown to work also with bacteria, analyzing bacteria. But it means that their evolution and their ecology and their epidemiology and the immune selection occurs all in the same time scale. So we want to actually start integrating these things, uniting these fields, and bringing them together so that we could study them jointly. Couple of assumptions. I do say that evolution is tree-like. It probably looks more like a banyan tree than it does a typical tree where everything branches between different areas. And I will use some of this tree analogy. So we have these color points here. These are leaves or tips. They're also the sampled viruses or the sequences. They are connected by branches. The length of the branches tells us how much time has passed or how much genetic information has changed over that period, and they come together in nodes. Just like you were describing a tree, tree branches come together at nodes. And the tree branches go all the way along a trunk to a root, okay? Trees go from the root, that's the ancestor when we look at this tree. So we'll say the oldest part is the tree of the root, and it grows forward to the leaves. An important thing about phylodynamics is that viral genomes they're independent measurements of that epidemic process. So it doesn't replace what we're doing by collecting samples, counting numbers of infected individuals, and those population scale modeling that we're doing. This is an additional set of data that doesn't replace that previous work, but actually complements it very well. And finding a way to unite them is a really big challenge. So here's a nice example. We build a tree and we'll say, okay, let's create a hypothesis and test it with that tree. So we're now looking at the global scale of influenza viruses, and we're asking the question, do we have the northern epidemic and the southern epidemic mixing together along the phylogenetic tree, like we see in A, which would be a migration, tell us that there's migration between these epidemics? Or do we see something where we have the northern epidemic is independent of the southern epidemic, and that they may show up seasonally, but they're not mixing? If we understand what these two models look like, they'll tell us something about the control effort. Well, when we do that, here we have Eurasia, New York, Japan, Hong Kong, Southeast Singapore, Australia, and New Zealand, and we see through the tree, we get little pockets of individual locations, but they're actually mixed through the whole tree, okay? So we have a migration model, all right? That means that the viral population is moving between these different outbreaks and these epidemics. But what we don't know is there a global source? This is a fundamental question that people were trying to understand. So it says, even though we have these underlying migration between these different groups, we do have local epidemics. So in 2008, Rambo and colleagues said there's the epidemic timing was offset between the northern hemisphere and the southern hemisphere, but there was largely direction that was going between these two different epidemics, and it was probably coming from one place. So they argue that there's a single global source, and they argue that that source was in the tropics. They had limited data, but at that point it was the most data that we had available. They analyzed the seasonal dynamics in these different regions, and they said that it was coming from this one place, but that was based on an absence of data. So they didn't actually have samples from the tropical region. They said they don't have anything there, but it's coming from someplace. It must be where we haven't looked yet. Another paper that I think came out the same day, one in Nature and one in Science, from Colin Russell and colleagues, said actually the source is located in Southeast Asia and East Asia. And they said it was in that specific location. And they based their analysis, they said it came from this location, and they used antigenic and genetic data to base that conclusion. And they said that this region right here, was just a little bit more what they called advanced. But their fundamental assumption, well, red just means trunk, blue means, let's say that's northern epidemic, and green is southern epidemic. This is just a toy tree, so the words don't matter anything right there. But basically, if the virus that's found in the red section, we're saying that's the source population, and we're saying that from the source you get an epidemic in the north, and then you get an epidemic in the south. Okay? So if this is the pattern that we're seeing, we want to try and find out what's the location of this trunk on the tree. So if the trunk is located in a single location, that means that's the likely source. Russell said it was in Southeast Asia or East Asia, basically by calculating how far things were from the trunk. And they had better data than the Rambo study, but it was very qualitative. They just looked at the tree and said, this is closer. This must be where it's from. A paper that came out later from Trevor Bedford and colleagues said, actually, it looks like the population is just moving around. But again, they took all of Asia and called it, all of Southeast Asia and China, and said that's the source region here. And their model forced the connection to be, there had to be a source population in their model. So they said, okay, it looks like it's in China and South Asia. So we wanted to ask that question, and I just happened to be living in this part of the world at the time. I was living in Hong Kong. We had viruses that we had collected, so we said, hey, let's actually analyze that. And we made some assumptions. We said the virus is going to behave the same in all humans. Wherever they are, if humans are infected with influenza, it's going to behave the same. So we're not going to have to make any accounts on the virus mutating differently in different populations. And then we said the only important thing was that where they were found, not the size of the population that was being infected or the size of the epidemic. We didn't account for any other things that might actually determine whether those pathogens were being sequenced. So what we saw was that we found these coinciding epidemic peaks. Here we have Japan, New York, Europe, primarily Germany, US and primarily New York, and then Japan, Hong Kong, Singapore, New Zealand, and Australia. And we saw the ones in the northern epidemic, the northern region, their epidemics coincide. In the southern epidemic, or southern hemisphere, their epidemic peaks coincide, offset by 6 months. This is what we knew. Northern epidemic happens in the northern hemisphere during their winter, and then the southern hemisphere offset by 6 months in their winter. What was interesting was that in Hong Kong, we had two peaks. We had a peak that matched the northern epidemic and the southern epidemic. And then in Singapore, we basically had a flat line. We didn't actually see many oscillating peaks. And the dynamics of the outbreak in Singapore suggested that there were people getting infected, but there wasn't much transmission locally there. So it was very, very different epidemic dynamics in each of these different locations. So we created a model, and we said, okay, we can start adding time into our model and specifically test whether things went from tropical region to tropical region. If they're just transmitting within those tropical regions, then that supports it being a source population. If it goes from northern to tropical, tropical to southern, southern to tropical, then it says that we have this metapopulation that's moving up and down through these different regions. And when we did this, we had to make some assumptions, and we said, all right, there's a location that was defined by a season, so that said New York collected between 2002 and October 2002 to March 2003. That was a single season. That was called New York 2003. Influenza had that really strong temporal signal that I showed. It had that spindly tree. So it meant that we couldn't actually have time travel, right? We meant that 2013 could not be the source of 2003, okay? So we didn't have to turn off those transmissions in our model because they were highly unlikely to begin with. And we used a non-reversible, so now we can work out direction, saying, is it going from the northern hemisphere to the southern hemisphere? Is that the same as going from the southern hemisphere to the north? Okay, so we had some directionality in our model, and we wanted to know where the tree trunk was. Well, the tree trunk moved, okay? Going back to that first assumption, the tree trunk was going from Japan to Singapore to Hong Kong to Europe. It was moving throughout these different locations. So there was no single source population. And all locations occupied a big part of that backbone. Now, we don't know why it was one location or another, but it was moving. That tells us that we don't have a source sink population. We actually have a metadynamic population. And when we did our specific hypothesis test, as you can see, it doesn't just go from tropical region to tropical region every year. It's going between, it's going to the north, it's going straight from the southern hemisphere into the northern hemisphere. It's going from the tropical region into both north and south, and it was bidirectional. When we added more complexity to this and started breaking things down, we started seeing a much more clear picture of how this virus was moving. So it wasn't one location, it was a large dynamic population that was moving between locations. Okay, and I think I've pretty much said all of this. That we had these epidemics that were moving constantly, no single source, and there was really the fact that we didn't see seasonality in the tropical region was, that just might be artificial of what we did, or it might be that there really is no seasonality in that model. One thing that I will emphasize is that this study still has no information from India. It has very little information from Africa and little information from South America. But we say that we don't have a source sink model, so we don't have a global source. This is important because if we know where the global source is, then we can stop influenza. If it's in one place, we could target vaccination, we could eliminate that disease. That is not going to be a strategy for eliminating influenza. When people have done more recent work, we see that actually the major driver for spread is how many people are moving in airplanes between locations. Viruses don't move, people move. So it makes sense, we look at passenger flow, and that becomes a major predictor for how viruses move around, how many people move between these different locations. The other element that came out of this was that actually age matters as well. Children don't travel as much as working adults. So maybe we need to focus our vaccination strategy at the global scale on those people that travel. But if we consider each population as a somewhat isolated network, we have a globally connected network that are connected through flights, and we have epidemics that coincide in the north and coincide in the peak. And these are spatially separate epidemics. There might be migration between these populations, and we only detect it during those seasonal peaks. A place like New York is extremely well connected, and it may be that those epidemics in that location might be the source population just because of how well it's connected. But we still don't understand why. We suspect that it might be epidemic size as well as global flow, but it's not entirely clear. And transmission into the Southeast Asian region might be crucial for transmission to the south, that we might need to have these these transmission events to support the global network, but that region is not the source population. Okay, so that's where we are for influenza. That's the foundation of a lot of this work at that global scale. Let's now extend it to RSV. Well, RSV shows similar dynamics. This is an analysis of one of the genotypes, ON1. It emerged, it was detected in Ontario, and it rapidly spread around the world and replaced other genotypes, okay? And it's found all in these different places, and we see it's also got some global migration dynamics. The difference with RSV, though, is that it doesn't travel as fast, and it can persist in one location for several years. So we do have some similarities, but we have some key differences. But using the same method that we used with influenza, we started interrogating RSV. Now let's go down to a lower scale. I'm now going to take that genetic data and try and understand HIV transmission within the city of Houston. This is to sort of give you an idea about some of the methods that I'm going to introduce and expand on in the next section with RSV and SARS-CoV-2. But starting here with with HIV transmission within the location. Within Houston, We have a wide economic difference between the populations. We have many people that are undocumented, that resist access to health care, and they're not necessarily being enrolled in treatment programs. And then we have a population that speaks multiple languages that may not be getting access to information. I'm sure a lot of these sound similar, and they are challenges that we're not necessarily dealing with well within the U.S. But when we just take the genetic data for all those infections within the city of Houston, these are all different individuals that have been sampled, tested positive, and sequenced over many years. When we look at how genetically similar those pairs of sequences are, we can start identifying individual pairs, likely transmission between two individuals, or clusters, where there might be a large amount of transmission between multiple individuals in that population, and that can go on for several years. So we want to understand the characteristics of this population. Is it race, ethnicity? Is it birth year, right? Can we identify what part of the population is driving the spread within Houston? So we had this information, working with the Houston Health Department to try and interrogate the demographic features and link it to transmission events within the city of Houston. When we did, we could identify those transmission clusters, and we could build a whole bunch of little small phylogenetic trees that were linked with missing information. So we don't know what's happening on these black branches, but we know what's happening within these different groups. So now we have this many phylogenetic trees, however all these different groups are. And from that, we wanted to just model just those demographic characteristics averaged across all the populations. So we're not creating just one model. This is our model, that it goes between red to yellow or yellow to yellow, between a firefighter and a waiter, or to a musician, or to a executive, right? So we don't know how it's moving within these groups, but we said, okay, these are the characteristics, and let's put that in the model. And we have one model that's fit for all of the hundreds of trees that we had, okay? So we're not doing just one model, one tree, we're doing one model for every tree, okay? And it's this averaged approach. When we did that, we saw actually there was a race ethnicity. So it was transmission between Latin Hispanic individuals, and it was directional, Latin Hispanic to Black American, and they were being infected from another group, the white ethnic group. So we had some structure in the transmission within that population, and then we also had a little bit of age structure going between people that were born before 1990 and people that were born after 1990. So we had this population split between these two age groups. We saw that there was some transmission in age group, but more importantly, there was race ethnicity structuring. So they could target the population with proper language and accessible information to provide them with information they can use to stop transmission. So that's where we start at the local scale. Now we've got more data than we've ever seen for any single disease. And we want to start analyzing that and looking at transmission within a specific region. Everything I've told you before, none of those methods could handle what we were dealing with SARS-CoV-2. So now we had to come up with new methods. A PhD student within my group jumped at this, and he said, well, if I can characterize this entire tree, then maybe I can just color a branch and determine whether it's a locally circulating branch, whether it's a branch that has been introduced from another location, or whether it's a branch that's going to another location. So is it something that's been imported, locally circulating, or exported? And that's all these colors mean. And when we do that across the whole tree in this small period, we can start determining how much of an epidemic is spreading to another location, how much of an epidemic is circulating locally, and how much of it is actually because we're seeing introductions. And that tells us whether that population might be a source or a sink. So when we looked at just the state of Texas and that transmission within the state of Texas, we saw, yeah, there was introductions from other locations, but most of the epidemic was circulating locally. So we did get some introductions globally or domestic introductions, but really the epidemic, when it became an important outbreak within Houston, it was circulating locally, and it was circulating locally quite quickly. When we look at imports versus exports, within Houston, we had few imports. We had more early on, but most of it was locally circulating, as I mentioned. When we compare it to a smaller rural region, we had almost no local circulation. All of it was being driven by imports. So you need different control strategies for the smaller rural, less densely populated region compared to the highly densely populated rural urban regions. And we had this, we could identify these 4 or 3 locations across the state of Texas that were the major sources. So now we're narrowing in that scale. We've gone from that global scale. Using that genetic data, we can now go to within one location, and we start seeing, okay, Houston's a major source, Dallas is a major source, and San Antonio, Austin was a major source. These are the largest urban regions within the state. But all the other places that were smaller, they were dependent on introductions, and their primary introductions were coming from these urban regions. They weren't coming from outside of the state. That now gets us down to the state level. Getting down to an individual city requires even further data integration and new methods. Okay, so we see now that there's differences within a single location, but within that state, that is really tough. And the only way to do this is to partner with those public health people and those individuals that are working with those communities. Okay, you need buy-in from the community, and you need buy-in from those researchers that are collecting the data. You need things like electronic data records. And you need a set of permissions that allow that information to be shared between people. These are huge problems that I don't think anybody has found a good solution to. But I know that I needed to get a massive set of permissions for us to look at the data that was collected by the Houston Health Department, which was next door to our research building, right? So proximity wasn't the problem, but really there was a set of structures in there, and nobody knows how to deal with that information yet. And that scales up to everything. In Europe, going between countries. In the US, going between bureaucratic organizations. Nobody knows how to deal with sharing data yet. So if you guys have some ideas, please tell me, because I hope I can take it to these other groups. But once we find a way to share data, then we can start doing some hypothesis testing and some more statistical modeling. So, like I said, we needed new methods. We couldn't just use that pairwise comparison to identify those separate groups. We actually had to come up with a new way of identifying clusters. So, again, this is Luke Liu. He's published this work recently in PNAS. He found a way of traveling across the tree, looking at whether a branch was locally circulating or introduced, and by moving between these different branches, identify clusters that were locally circulating versus introduced events. And it was able to work on data sets that were, I think in this one, it was about 25,000 SARS-CoV-2 genomes. Okay? So now we're analyzing a larger data set than we've ever looked at, and we're moving down to the scale of a local scale, which is how we might be able to stop transmission locally. That is where we have impact on using this genomic data. Everything I've told you previously, those genomic data makes us really pretty pictures and gives governments ways to blame other governments for causing an outbreak in their location. But it doesn't really give us really effective tools to stop it. This will give us effective tools to stop an outbreak within a location. So we have that large tree, we identify these smaller groups, by defining each of these different nodes, whether it's come from one location of interest or it's come from a different location, and whether that branch has changed from something that's circulating locally versus being introduced. When we did that, we were able to take our tree that had 25,000 genomes or so down to, I think, about 150 small trees that could be analyzed and that we can make some inferences on. And then when we did that, we also had to come up with a new statistical method. So we developed this method called the tip-trace association test. It's actually, we didn't develop the tip-trace association test. That was done by another individual, Joe, I don't remember what his name was now. Sorry, the program is BATS. But we took that inspiration and we developed it further, the tip-trace association test, that we can work on large trees and have a better idea of what significance was. So we developed that test a little further so we could start correlating age, gender, location with the structure of the tree. And when we start looking at things, we said, okay, is there some sort of segregation by gender? No, there wasn't. But there was segregation by age for these different populations. And larger clusters showed that age was a really big determinant of the spread versus smaller clusters. Really small clusters could have, we didn't have any signal of age in that transmission. But larger clusters were really determined by age. And what we saw was middle-aged adults, individuals that were saying, I'm way too important to stay home and stop transmission. I'm opposed to wearing masks. For whatever reason it was, if they were adults, they were more likely to transmit, and they were transmitting to those high-risk groups, those seniors, as well as infants and children. So that was a major bit of information that this was, the young adults were a major source population, young and middle-aged adults in there. The ones that should know better, by the way, I'll say, ones that can take that information, process it, and stop transmission, they were the ones that saying, I'm not going to do that. And they were infecting other people within their communities. When we did look at transmission and those age structure, we saw that within Houston and Harris County itself, it was transmitting to these other locations. But surprisingly, there was transmission between these locations, looked to be driven by school district. Outbreaks within the location were often clustered by school district, but the connections between locations were associated with adults moving between them. So we saw children and teenagers are often in daycares or in schools. They're not really moving. That's where their exposure was happening. But adults had the least constrained movement, and that was driving the spread within that local community. Okay. Now, like I said, we're trying to go really fast here. We've gone from the global scale all the way down to outbreak within a single location and looking at transmission. But we do have a number of epidemics that coincide. We treat influenza like it's in a vacuum. We treat SARS-CoV-2 like it's in a vacuum. But actually, influenza A, H3N2, influenza A, H1N1, influenza B, Yamagata and Victoria lineage, and RSV A and B co-circulate. Those are 6 separate pathogens that co-circulate, that have similar epidemic peaks and have similar epidemic dynamics. In this study, we don't have SARS-CoV-2 because the data set we were using was older than SARS-CoV-2. But that's another one now that is co-circulating, right? It hasn't gone away. So we have these epidemics, they co-circulate, and we treat them like they're unique and they're something special. We can ignore RSV because we're focused on influenza. This is this idea that we need to get out of our single ivory towers and our disease focus, and we need to actually start focusing more on syndromes and systems that might be distorting our view of individual health and population health. Again, back on my soapbox. But when we have these different viruses, RSV, influenza A, and influenza B, they have coinciding epidemic peaks. Within the US, when we look at all these separate regions, when we look at the whole scale, it looks like these things are entirely in sync. When we start looking at these things at the specific regions, they can be offset by a couple weeks, right? So you can have an epidemic peak. It's influenza A, it's influenza A, it's influenza B. Then everybody that shows up gets RSV, and you get no influenza A for a few weeks. And then we get influenza B. And at that national level, we don't see that. But in those local scales, we do see that. Individual doctors will sit down and say, no, it's not influenza A right now, it's all RSV. Everybody that shows up has RSV, right? And they understand that. But at that global scale, we are missing that signal entirely. So we wanted to look at multiple pathogens within a single location. When we did the phylogenetics, we start seeing that, yeah, we have these multiple peaks. They're all matching up. We know that these things are co-circulating, and we're able to separate out these different viruses. But it does lead to a problem that we don't actually know how to do this. So we create a simulation. Within this model, I'm not throwing up all the mathematical formulas, but if you go across one row, that is a single virus that says, is an individual susceptible? Have they been infected? Have they now recovered from that infection, but they have some sort of cross-protection or cross-immunity phase? And then do they become fully recovered and they're no longer part of that epidemic for that region? So there's a period here where we say this XCS, right? That's where they are susceptible to the other disease, but they are cross-protected in some way. We don't know what that is. Or we say, is there something preventing, if you're infected with one disease, is there something preventing you from being infected with another one at the same time? So we are able to look at these two parameters. Is there an inhibition of infection, or is there some sort of cross-protection? Without going into what the biology was, we just wanted to look at the pure numbers. When we did that, for RSV and influenza A, we saw that actually we have some sort of cross-immunity and some sort of prevention of co-infection, inhibition of co-infection, that allowed us to, if we account for that, we more accurately recovered those epidemic dynamics than we saw if we just used the pure count data on its own. And when we compared RSV to influenza B, we had some sort of cross-immunity. It could be that a person that's infected, once you're infected, your innate immunity just says, we don't want to get sick again, let's throw up all the alarm bells, and it's somehow preventing infection to come in. It could also be that somebody says, my kid was sick, I heard there's an outbreak, I'm going to reduce exposure, right? We don't know exactly what that is, whether it's biological or epidemiological, but there's something that's stopping those epidemics from really coinciding. Okay, so to do that, the analysis does support that there's some sort of competitive interaction between RSV and seasonal influenza. We speculate that cross-immunity after infection might be a major driver of this viral competition. And this was really led by this young woman here, Jiani Chen, and developing this work. Oh, this is actually, sorry, it's not in review. It's been published in Epidemics. Everybody there is signing. This is W's. If you want to say wow in sign language, your mouth becomes the O. Wow. So that was our conclusion from that. Okay. So I'm going to try and wrap all this up. Remember, I crossed a lot of scales. Like I said, we'll start slow, but it was going fast. So please ask questions, follow up. But there's a lot that we went through today. So genomic data itself is central to our modern approach to infectious diseases. Okay? We might be focused in a hospital or diagnostics. I've said this to a number of people. As a clinician, if you're given a viral sequence, that does not change your treatment plan than if you're just told it's a virus. right? So you don't actually need the sequence to improve treatment. So it's not actually effective at that scale. But at the population scale, it's becoming very central. And some of the earliest information that we learned about SARS-CoV-2, the H1N1 pandemic, the emergence of Zika, all came from genomic sequencing. So it is important to our public health scale right now. And we're also in an area where we have the capacity. We've stopped doing it, I think because people got a little fatigued from SARS-CoV-2, but we demonstrated that on a global scale, we could come together, generate genomic data, and share it. So those barriers that are preventing this sharing of information are artificial, and we can do it. And it takes a monumental effort to actually do it, but we can. And I think I like to advocate for academic and public health partnerships, that we can do things in the academic realm that'll tell us information, but if we partner with people that are working with populations, we can actually make sure that these solutions and the information that we learn is translating to stopping disease and disease spread within communities. And I think whether you're an academic or you're a clinician or you're working at these global scales or individual scales, ultimately that's what we want to do. We want to stop disease transmission and improve the health of the populations. So I think that's going to be key moving forward. And then these challenges exist to make sure that everybody actually sees the value of it. We can't just focus on an individual country that does this work, or we make sure that we develop a method, but we don't provide support for it being used in public health and used regularly. We could break that down. So we have to make sure that all stakeholders actually realize that value. Okay, I'll close it up there. Partnerships with the Houston Health Department and the Georgia Department of Public Health. Lots of brilliant collaborators. This group here within the Centers for Disease Control have been wonderful advocates for this program and trying to move it forward, as well as this group within NIH. So you guys have the Emory SEER program here. I was part of the St. Jude SEERS program. Now I'm part of the UGA SEER program. So we're competitors. No. And other programs that have continued to support this effort. I say thank you.
So since this is an oration, we give it to the speaker if he is willing to accept a few questions. We have about 5 minutes for questions. Are you okay?
Yeah, I'm quite happy to take questions. Yep.
Questions from the audience, anybody?
Yeah.
John? Okay, so let me begin with one.
Okay.
Now, it's a case of too much, lots of sequencing, lots of, in an epidemic, everything's going to be the same. So what's your bare minimum number that you would want as an ideal, as opposed to what is the reality? And how do you reconcile with that, with the scale of how much is generated?
Yeah. So for SARS-CoV-2, we estimated we needed transmission of about 2 to 3 weeks for us to actually see changes within the population. Okay, that was a chain of about 4 people. If it was just a sequential, right, in the period of exposure, infectious, and then recovered. So we needed a time frame of about 3 weeks. Okay, now that's how we knew we had basic information there. The other thing we found out, though, is that if we had those things with zero information, there was additional things like you had no change, but they were from 2 kids that were in different school districts. So the zero information actually became really important as well. I think we've got some new tools. AI has been really good at sort of helping us design data sets. AI is a black box and will always give you an answer, right? I want to warn everybody, don't believe it just because it gives you an answer, right? Because I believe in statistical approaches, right? Statistics tell us what's important. AI has been really useful in saying, here's how you can manage this data. And I think that's a good thing that we have there. So I'm not going to say we only need 100 sequences, because if we can get 1,000, we might be able to uncover something that we didn't even know about those outbreaks. And I think there's value both in the actual changes and when they're identical, but we need to be able to tie it in with those characteristics of the population, right? The sequence on its own is not valuable. The epidemiological demographic data on its own has limited utility. Combining them together, I think we're finding new ways to learn about it. So that was a good way of avoiding your answer, or when you answering your question. Yeah.
Brilliant lecture on genomic surveillance and its importance. But I was particularly fascinated by the co-infection. Early in January 2020, when I was in Pune, we were taking specimens and looking for influenza and RSV along with SARS-CoV-2. And people were not ready to accept that co-infections happened. So I think that information can be patchy depending on whether people looked for it at that point in time.
Yeah.
And also how symptomatic people were, because people are yet getting used to the idea of surveillance.
Yeah.
Which is, in good times and bad times. So, do you have any take on that?
So certainly there was a lot of sensationalist headlines, right? Warning, the syndemic is coming, right? And I think it's really important to look for it. I mean, I don't know, can you tell me maybe how much you found co-infection?
I don't know.
You don't, okay.
Cumulative effect of SARS-CoV-2 and
Yeah.
RSV and influenza. What would that do to an individual?
Yeah, from the individual level, so we found, I think between 1% and 3% had some co-infection with those viruses. There were co-infection with other things might be a little bit higher, but those ones in particular was quite low. So I don't know how important co-infection for those particular viruses are going to be. I think the co-infection, when you get co-infection with bacteria and co-infection with other things that sort of operate on slightly different organs, are going to have a bigger impact at that individual level. I know that it complicates treatment, right? Like if you're trying to deal with a viral infection and then you get a secondary bacterial infection, or the other way around, it might be masking the actual cost. At that population scale, I don't think we have a good idea as to what the cost is. Even if people get a little bit sick, or maybe sicker than they would, but they don't actually go into the doctor, or they get treated for one, I don't know if we really know. I think you're right. People aren't prepared to actually look for it, and I don't know if we have good methods to look for it yet. I think like I said, there seems to be little current value at the clinical stage to say, oh, here's the sequence of influenza. But knowing that there's influenza and SARS-CoV-2, I don't know how that might change treatment, but I think the potential impact is definitely there. I just don't know what that is yet. And it might be a big impact on a small section of the population. Like maybe it's a big impact on less than 1% that actually get co-infected. So I don't know how to answer that in a good way. Yeah.
Can I ask you the question that's echoed again and again in people's minds? Based on your expertise, what is the source of SARS-CoV-2?
Thank you for putting me on the spot, and this is now being broadcast. I think when I look at the evidence suggests that it's a natural emergence from the market system. That doesn't mean it couldn't have been a accidental leak. I think that there's still a chance that it could have been, but I think it's the product of a natural process. It has not been engineered, right? I don't think there's any evidence to say that it's been an engineered virus or this is some sort of gain of function that has led to a leak. But I think the data seems to lean more towards natural spillover in the market system, but it could have been a leak of a natural virus that somebody collected. I'm getting good at not answering questions, right? I lean towards a natural emergence. I do.
Thank you for such an engaging talk. I think I can sit here for all eternity listening to you. That as a credit. My question to you is, probably, looking at the phylodynamics, I understand a little bit, okay, but with the astronomical number of, this data that you showed, what I'm keen is, for example, how accurate one could be in terms of forecasting or predicting it? Does it have a predictive power? Because what you've shown is somehow, sort of fits in with a priori, information that you have. So I just want to understand that, and then extension of that from your response.
Yeah. So we were doing some work on forecasting from Phylodynamics for some other projects, for avian influenza and for other things, and I think it's got really good potential. I think understanding how this reverse time simulation, how things have occurred in the past to generate our current understanding, is going to allow us to make more accurate predictions going forward, right? So I think that there's a lot of potential, but I think the work that's been done right now on forecasting evolution and forecasting epidemics is still really in a very early stage to do it well. We've been trying to predict the next influenza seasonal the next seasonal dominant strain, right? That would be like the holy grail. If we could tell you that based on what I've seen today, that this virus is going to dominate, that would mean we would have better vaccines. And if we could do that, that would be amazing. We do it really well for forecasting about 2 weeks ahead. We do it really well going up to maybe a couple months. It's within error, but it starts breaking down as we go further and further ahead. So I think we're still in a very early stage of forecasting. I do think we're going to get there, though. And I would say 5 years ago, I would have said, I don't see the value in it. I would have leaned, I said, I wanted to understand more about what was happening in the past to describe the phenomenon I observed today, rather than trying to predict what might happen in the future. But I do think we have some new tools and new approaches that are showing really good promise.
The second thing is probably something related to that, but then probably you have to take a prophetic role. What do you think, after you analyze so much data on SARS-CoV, would you think in the future it'll be sort of attenuated, or do you think that it would just, the villains will become more? That is what I need to understand.
So that's a tough one. So so if we look at the 1968 influenza H3N2 pandemic, right? That had a global impact and killed many people. But it's circulated since 1968 seasonally and continues to circulate. It's probably killed more people in that seasonal period where we might argue that it's attenuated than it did during the pandemic, right? So the impact, right, I don't know if attenuation is the right way to describe it, because clearly it's not maybe killing as many people that are being exposed to it, but it has a cumulative impact that is much larger than the pandemic. And I think SARS-CoV-2 is probably going to be in that situation. As long as it continues to circulate, we're always going to have susceptible individuals within our population that are going to be exposed, and that over the long run, it probably will have a larger impact than it did during the pandemic, which I've never seen anything like that in my lifetime. So that's the largest single casualty event that I can think of in my lifetime. So thinking about if it circulates another 20 years, it could be much larger, yeah.
A last question. In the opening of your talk that you showed 3 trees that are different with seasonal and other 2 viruses.
Yeah.
Now, for example, in your reckoning, you find some sort of a punctuated event that would have, actual cause of, decided the evolutionary trajectory, or it is always some sort of an accumulative mutations that you've observed?
So I'm not sure if I heard all of that question. I may have lost a little bit there, but I think you were saying, do we see punctuated changes, or do we see accumulated over time?
Yep.
Okay. Again, I'm not going to give you an entire answer, because I think in different parts of the tree, we see both, right? So in seasonal influenza, we have a really clear accumulation and changes in the genetic tree. But if we look at the antigenic data, which is how we collect vaccines, and linking genotype and phenotype is probably the largest challenge that we've never actually successfully or actively bridged in molecular genetics, right? So we have the phenotype of antigenicity, and we have genetic evolution. The genetic evolution looks like it's much more regular, accumulated over time. The phenotype says, well, we need this vaccine today. Here's all this group. It's Victoria 68 or Hong Kong 68, and then it jumps to another area, and it says, okay, we need a new vaccine strain. And then it jumps to another area, and we need a new vaccine strain. So the phenotypic evolution is punctuated. The genetic evolution is gradual. So again, it's really hard to sort of entirely work out. Yeah.
With that, thank you, Justin. That was lovely. Can I call upon Dr. Jacob John to hand our speaker to read the citation and to also give him a memento from our side?
The Fieldhouse-Myers Oration has been instituted in the memory of Jeanette Fieldhouse and Dr. Ruth Myers. The late Dr. Ruth Myers was a close friend of the former and was an eminent researcher in the field of microbiology and virology. Dr. Myers has done pioneering work in this field at the Christian Medical College, Bahl. This citation is in commemoration of the 14th Fieldhouse-Myers Oration for the year 2025, delivered by Dr. Justin Bahl from University of Georgia, Athens, on the 8th of December 2025 at the Christian Medical College, Bahl. The honor is in recognition of his extraordinary contribution to advancing pathogen genomic surveillance, statistical, phylogenetic analysis, and actionable public health intelligence in the field of respiratory viruses. CMC Vellore is proud to present to you, sir, this.
Thank you, Justin.
Thank you for the wonderful talk.
Great. Thank you.
And last item today, just a vote of thanks. A big thank you to all of you who have gathered here, and most of all, a big thanks to Dr. Justin Bahl, who's traveled all the way from Georgia, long flight. Want to take a moment to thank all the participants all the members of the department who have helped with the organization, Mr. Divakar, Mr. Prasanna, who have helped with sending out the letters, the secretarial staff, and a big help to Dr. Ramit, who was able to travel to Chennai to bring back the speaker to Vellore. Thank you all for being here, and I hope you enjoyed this academic feast. Thank you.
This is for my kids.
You want to take a photo?
I'm going to